On and start on a new topic which is called activation of T lymphocytes. This is chapter 9 in the book of cellular and molecular immunology by Abbas and others. So in this chapter we'll see in the overview of T cell lymphocyte activation, signals for T lymphocyte activation, recognition of antigen, role of co-stimulation in T cell activation, functional response of T lymphocytes, changes in surface molecule during T cell activation, cytokines in T cell activation, clonal expansion of T cells, differentiation of activated T cell into effector cell, development and properties of memory T cells, then you see the decline of T cell response, then you see the summary. So let us go. The process of T cell activation generate from a small pool of naive lymphocyte specific for antigen, a large number of effector cells with the same specificity that function to eliminate that antigen, and a population of long-lived memory cells that can rapidly react against the antigen if it is reintroduced. So a fundamental characteristics of T response, like all adaptive immune responses, is that it is highly specific for the antigen that elicits the response. Both the initial activation of naive T cell and the effector phase of T cell mediated the adaptive immune response are triggered by recognition of antigen by the antigen receptor of T lymphocytes. In chapter 6, we described the specificity of T cells for the peptide fragments derived from protein antigen, which are bound to and displayed by self-major histocompatibility complex MHC molecule. In chapter 7, we described the antigen receptor and other molecules that are involved in the activation of T cells by antigen and the biochemical signaling initiated by these receptors. In this chapter, we will describe the functional response of T cell. We begin with a brief overview of the T cell activation, discuss the roles of co-stimulators and other signal provided by antigen-presenting cells, the APCs, in T cell activation, and introduce the sequence of proliferation and differentiation that occur when a CD4 plus and CD8 plus T cell recognize foreign antigen. The generation and functions of effector CD4 plus and CD8 plus T cells are described in more detail in chapter 10, 11, and thus chapter 9 and 10 and 11 together cover the biological function of T lymphocyte and their roles in cell-mediated immunity. Overview of T lymphocyte activation. In immune responses, T lymphocytes have to recognize. Just you can follow me, please. Just follow me. I cannot put everywhere the cursor, but you can follow me. So I'm repeating. Overview of T lymphocyte activation. In immune response, T lymphocytes have to recognize the same antigen at two stages. First, to initiate the response and later to perform effector function. Antigen activate naive cells to proliferate and differentiate into effector and memory cells. Then the effector T cells are activated by the same antigen to perform the function that lead to the elimination of the source of antigen, infected cell or tumor. As we discuss later, the requirement for these two activation events differ in terms of antigen presenting cells involved and other signal needed. The initial activation of naive T lymphocyte occur mainly in secondary peripheral lymphoid organs, through which these cells normally circulate and where foreign antigen are concentrated and presented by mature dendritic cell, the DSC. See figure 9.1. Clones of T lymphocytes, each with a different specificity, are generated in the thymus before antigen exposure. naive T lymphocytes, which have not previously responded to antigen, circulate through the body in their resting state, and they acquire powerful functional capability only after they are activated. The activation of naive T lymphocytes occur in a specialized region of lymph nodes, spleen and mucosa lymphoid tissue, where naive T lymphocytes interact with the dendritic cell that have captured antigen from tissue or blood. The antigen recognition, together with other activating stimuli, induces several biological responses in T cells. Cytokine secretion and increased cytokine receptor expression, proliferation, leading to an increase in number of the cell in the antigen-specific clones called clonal expansion, and differentiation of the naive cells into effector and memory lymphocytes. The process of the T cell activation is associated with changes in the expression of numerous surface molecules, some of which are involved in a trafficking of the T cells, and other plays important roles in inducing and regulating T cell response. Antigen-presenting cells not only display antigen but also expresses surface molecule and secretes cytokines that affects the magnitude and nature of T cell response. The role of antigen-presenting cell in instructing T cells how to respond to different categories of pathogen are discussed later in this chapter and in chapter 10. So there are a number of diagrams that you can see, and I will not want to go through. If you really wish, you can go and see what is happening. Even if I go through, I cannot explain everything. Maybe just let you know the figure. What does it explain? This is figure 9.1, activation of naive and effector T cells by antigen that are transported by the dendritic cell to lymph nodes are recognized by naive T lymphocyte that is recycled through these lymph nodes. The T cells are activated to differentiate into effector cell, which may remain in lymphoid organ to help B lymphocytes or migrate to site of infection, where they are again activated by antigen and perform their various functions such as a macrophage activation. So the dendritic cells, they act as the antigen-presenting cells. They always go back to the lymph nodes. So here you see the dendritic cell is carrying the antigen, and it is presenting it to some of the T cells, the effector cells. This is figure 9.2, sequence of event in T cell response. Antigen recognition by T cell induces cytokines, Interleukin-2, secretion, particularly in CD4 T cell, clonal expansion as a result of cell proliferation, and differentiation of the T cell into effector cell or memory cell. In the effector phase of the response, the effector CD4 plus T cell respond to antigen by producing cytokines that have several actions, such as the recruitment and activation of leukocytes and activation of B lymphocyte, while CD8 cytotoxic lymphocytes cell respond by killing other cells and secreting inflammatory cytokine. Antigen presenting cell and others they're working together. So we see they have the naive T cells, CD4, naive CD8 T cells all together. They just need to, when they're in lymph node, they are moving around, but in peripheral, they can go to peripheral after being activated by cytokines. They can differentiate to effector cell and go to the site of scenes for action. Let us continue. Proliferation and differentiation of T cell are regulated by several feedback mechanisms. For example, activated T cells deliver signal back to the antigen-presenting cells, further increasing the antigen-presenting cells' ability to activate T cells in a positive feedback loop. At the same time, some surface molecule expressed on activated T cells as well as a cytokine secreted by these cells inhibit further activation, and these negative feedback mechanisms serve to establish safe limit to a response. Effector T cell recognize antigen in lymphoid organs or in peripheral non-lymphoid tissue, and they are activated to perform functions that contribute to the elimination of microbes and in disease state induce tissue damage. Whereas naive cells are activated mainly in secondary lymphoid organs, differentiated effector cells may respond to antigen and carry out their function in any tissue. The process of differentiation from naive to effector cell gives the cells the capacity to perform specialized function and the ability to migrate to any site of infection or inflammation. At these sites, the effector cells again encounters the antigen for which they are specific and respond in the ways that serves to eliminate the source of antigen. Effector CD4 plus T cells, called helper T cells, can recognize antigen and microbes ingested by macrophage or B cell and activate these cells, and effector CD8 T cells can recognize antigen on infected or tumor cells and kill these cells. Upon antigen-induced activation, CD4 plus T cells secrete cytokines and expresses a cell surface molecule that can activate other immune cells. These effector T cells are classified into subpopulations on the basis of their cytokine profiles and function. Some of the CD4 effector T cells activate macrophage to kill phagocytose the microbes. Others secrete cytokines that recruit different types of leukocytes such as eosinophil and neutrophil, which destroy different types of pathogen, and yet others remain in lymphoid organ and help B cells differentiate into antibody secreting plasma cells and memory cells. Cytotoxic T lymphocyte CTLs, the effector cell of CD8 plus lineage, kill infected cell and tumor cells and also secrete cytokines that activate macrophage and induce inflammation. Memory T cells that are generated by T cell activation are long-lived and have enhanced ability to react against antigen. These cells are present in the recirculating lymphocyte pools and are abundant in mucosal tissue and the skin and in the lymphoid organs. After a T cell's response wounds, there are many more memory cells of the responding clones that there were naive T cells before the response. These memory cells respond rapidly to subsequent encounter with the antigen and generate new effector cell that can eliminate antigen. T cell response decline after the antigen is eliminated. This process of contraction is important for returning the immune system to a state of equilibrium or homeostasis. It occurs mainly because the majority of antigen-activated effector T cells die by apoptosis. One reason for this is that as the antigen is eliminated, lymphocytes are deprived of the survival stimuli that are normally provided by antigen and by costimulator and cytokine produced during inflammation reactions associated with infection and other types of antigen exposure. In addition, inhibitory mechanism activated by antigen recognitions function to control the magnitude and the duration of response. With this overview, we will proceed to discussion of the signal required for T cell activations and the steps in response to CD4 plus and CD8 plus T cells. We will conclude with the discussion of memory cells and decline of immune response. Signals for T lymphocyte activation. The proliferation of T lymphocyte and their differentiation into effector and memory cell require antigen recognition, co-stimulation, and cytokines. In this section, we will summarize the nature of antigen recognized by T cells and discuss specific co-stimulators and their receptors that contribute to T cell activation. Cytokines are discussed later in this chapter and in chapter 10. Recognition of antigen. Antigen is the necessary first signal for activation of lymphocyte, ensuring that the resultant immune response is antigen specific. Because CD4 plus and CD8 plus T lymphocyte recognize peptide MHC complex displayed by antigen-presenting cells, they respond to protein antigen. The natural source of peptide or in chemical that bind to and they modify protein, thus creating novel peptide. Protein antigen that cross epithelial barrier or are produced in tissue are captured by dendritic cell and transported to lymph nodes. Antigen that enters the circulation may be captured by dendritic cell in a spleen, as discussed in chapter 6. Both naive T cell and mature dendritic cells are drawn to the T cell zones of secondary lymphoid organ by chemokines produced at these sites that engage the CCR7 chemokines receptor on the cells. By the time the mature dendritic cell reach the T cells area, they display antigenic peptide on the MHC molecule and also expresses costimulators. Some soluble protein antigen in the lymph node may be derived to lymph nodes independent of dendritic cell and are taken up, processed, and presented as peptide MHC complex by resident dendritic cell within the lymph nodes. Dendritic cell present peptide derived from endocytosized protein antigen mainly in association with the class II MHC molecule to naive CD4 plus T cells, and peptide derived from the cytosolic and the nuclear protein displayed by class I MHC molecule to CD8 T cells. Naive T cells lymphocyte moves around within secondary lymphoid organs, transiently interacting with the many dendritic cell, and stop when they recognize the antigen for which they express specific receptor. T cells are in constant motion, mainly guided by a fibroblast reticular network, a matrix substratum produced by fibroblast reticular cells in the T cell zone of lymphoid organs. The dendritic cell in lymphoid organs adhere to FRC conduit are relatively immobile and simultaneously present many different antigens. The T cells move along conduit, making many successful contact with different dendritic cells. T cell recognition of antigen being displayed by these dendritic cells result in generation of biochemical signal that lead to rapid arrest of the T cell. This process stabilizes the contact between the antigen-specific T cells and the relevant antigen expressing antigen-presenting cell and allows the activation program of these T cells to be initiated. So A, you see an activated antigen-presenting cell costimulator deficiency. We see this one don't have costimulator event. Then T cell response, no response. If there is no costimulator, no response. The second, which is B, you see in that diagram, antigen-presenting cell activated by microbes in immune response increase the expression of costimulators, which you see from the antigen-presenting cell. You see the B7. So that one may cause a cell to differentiate, the naive cell to be activated and differentiate and as well proliferate and survive. So differentiated effector T cell can respond to antigen presented by cells other than dendritic cell. In humoral immune response, B cell present antigen to helper T cells and are the recipient of activating signal from the helper cells. In cell-mediated immune response, macrophage present antigen to and respond to CD4 plus T cells. Virtually any nucleated cell can present antigen to and be killed by CD8 T cells. Role of costimulation in T cell activation. The proliferation and differentiation of naive T cells require signal provided by molecules on antigen-presenting cell called the costimulator in addition to antigen-induced signal. The requirement for costimulatory signal was first suggested by experiment finding that T cells antigen receptor signal alone, this is induced by anti-CD3 antibodies that cross-link with T cell receptor CD3 complexes mimicking antigen, resulted in lower response than those seen in antigen presented by activated antigen presenting cell. This result indicated that antigen presenting cells expresses a molecule that works together with antigen for inducing T cell activation. These molecules are called co-stimulators, and the second signal for T cell activation was called co-stimulation. The first signal being antigen. In the absence of co-stimulation, the T cell that encounter antigen fail to respond, enter a state of prolonged unresponsiveness, or die. We'll see it in chapter 15. So the B7 CD28 family of co-stimulators. The best characterized co-stimulatory pathway in T cell activation involves the T cell surface receptor CD28, which bind to co-stimulatory molecule B71 CD80 and B72 CD86 expressed on the surface of activated antigen presenting cell. CD28 was discovered when stimulatory agonistic antibody against human T cell surface molecule were screened for their ability to enhance T cell responses when added together with the unactivated anti-CD3 antibody. This was soon followed by the identification of the ligand for CD28 called the B7 and later shown to be two homologous proteins named B7A CD80 and B71 and B72 CD86, often collectively called the B7. The essential role of CD28 and the B7 in T cell activation has been established by the T cell's immune deficiency caused by knockout of gene encoding these proteins in mice, and by the ability of the agent that bind to and block B7 molecule to inhibit T cell response in experimental animal and in human. The development of therapeutic agent based on this principle is described later. B71 and B72 are structurally similar integral membrane single-chain glycoprotein, each with two extracellular immunoglobulin Ig-like domain. CD28 is a disulfide-linked homodimer, each subunit of which has a single extracellular Ig domain. Its cytoplasmic portion contains several tyrosine and proline residue that are involved in binding of adapter and signaling proteins in the delivery of activating signal discussed later. CD28 is expressed on the vast majority of CD4 T cell, but in adult humans, almost 50% of the blood CD8 plus T cells, mainly effector and memory cells, expresses little or no CD28. This loss of CD28 is believed to be the result of chronic antigen stimulation, e.g. in chronic viral infection, and may be a way that T cells limit their own activation by the persistence antigens. Though this is the structure, you will see the structure. There is a B71, and we have it is a it's have the receptor to CD28, and inside the T cell receptor complex there is CD4 over there, and mostly we said it is mostly expressed in CD4 and almost 50% in CD8, but also it's less likely expressed in elderly people and mostly people who are having been constantly exposed to disease. So we continue. The expression of B7 costimulator is increased by microbial products and the innate immune response to infection, which ensure that T lymphocytes are activated only when needed. The B7 molecules are expressed mainly on antigen-presenting cells, including dendritic cell, macrophage, and B lymphocytes. They are expressed at low level on resting antigen-presenting cell and are induced by various stimuli, including microbial products that engage Toll-like receptor and cytokines such as Interferon Gamma, produced during innate immune reaction to microbes. The induction of costimulatory by microbes and by cytokines of innate immunity promote T cells' response to microbial antigen. This illustrates an important role of innate immune response in enhancing adaptive immunity. In addition, activated CD4 T cells themselves enhance expression of B7 costimulator on antigen-presenting cell by pathway dependent on CD40, described later, and this positive feedback loop serves to amplify T cells' response. Of all potential antigen-presenting cells, mature dendritic cells express the highest level of costimulators and as a result are the most potent stimulators of naive T cells. In Chapter 6, we mentioned the essential roles of adjuvant in inducing primary T cells' response to protein antigens such as vaccines. Many adjuvants are product of microbes or mimic molecular produced by microbes and necrotic cells and thus elicit innate immune response. One of the major functions of adjuvant is T cell activation. In T cell activation is to stimulate the expression of B7 costimulators on antigen presenting cell. An unactivated or resting antigen presenting cell in normal tissue are capable of presenting self-antigen to naive T cell, but because these tissue antigen-presenting cells express only low level of costimulators, potentially self-reactive T cells that seize the self-antigen are not activated and may be rendered permanently unresponsive. Regulatory T cells, which are important to tolerance to self-antigens, as discussed in Chapter 15, are also dependent on B7 CD28 mediated costimulator for their generation of and maintenance. It is possible that the low level of B7 costimulators that are constitutively expressed by the resting antigen presenting function together with the self-antigen that are displayed by these antigen presenting cells to maintain regulatory T cell. CD28 signal works in cooperation with antigen recognition to promote the survival, proliferation, and differentiation of the antigen-specific T cell. Co-stimulatory signaling via CD28 amplifies signaling pathways that are also induced down the stream of T cell receptor and may trigger the additional signal that cooperate with a T cell receptor induced signal. The PI3 kinase is recruited to the cytoplasmic tail of CD28 and this in turn activates the kinase AKT, which alter the cellular metabolism and promote cell survival. CD28 can also contribute to the activation of JNK, mitogen activated protein kinase via the RAC small G proteins and can amplify the activation of nuclear factor kappa B. The next result of this signaling pathway in T cells are the increased expression of anti-apoptotic proteins such as BSL2 and BSLXL that promote cell survival, increase the metabolic activities, enhance the proliferation and production of cytokines such as Interleukin-2 and proliferation of naive T cells into effector and memory cell. So these are diagrams which are important to pass through, but as I can see here, the expression of dendritic macrophage, you will see there is B7, 1 and 2. And the dendritic macrophage, B cell and others, there is ACOS ligands as a receptor. Then there is in dendritic macrophage, B cell, endothelial, epithelial, there is also PDL1 and other ligands for receptors. If you look carefully, then when you read this one, you will get the picture that the major members of B7 and B and CD28 families, the known B7 family ligands are expressed on antigen-presenting cells, dendritic cells, and macrophage and B cells, and CD28 family receptor are expressed mainly on T cell. Different CD28 family members stimulate and inhibit different stages and types of T cell response. The function of cytotoxicity lymphocyte antigen 4, CTLA4, and programmed cell death protein 1, PDA1, are discussed in chapter 15, and the role of inducible costimulatory ICOS, in the generation and function of T follicular helper cell is discussed in chapter 12. Other widely distributed molecules within homologue of B7, such as B7H3, B7H4, have been identified, but their physiological roles are not yet established. Other inhibitory receptors have also been identified, such as BTLA, TIM3, and TIGIT, but these are not homologous to CD28 and are not shown. Previously activated effector and memory T cells are less dependent on costimulation by B7CD28 pathway than are the naives. This property of effector and memory cell enable them to respond to antigen presented by a virus antigen presenting cell that may reside on non-lymphoid tissue and may express no or low level of B7. For instance, the differentiation of CD8 T cell into effector cell cytotoxic T cells requires co-stimulation, but effector cytotoxic T cells can respond to and kill other cells that do not express co-stimulators. Other receptors homologue to CD28 and their ligands homologue to B7 have been identified, and these proteins regulate T cell response both positively and negatively. Following the demonstration of the importance of B7 and CD28, several other proteins structure related to B7A and B7-2 or to CD28 were discovered. Some members of the B7 and CD28 family are involved in T cell activation, and others are inhibitor of T cells. In addition to CD28, the costimulator whose function is the best understood is ICOS. Inducible costimulator is CD278. Its ligand called ICOS-L, CD275, is expressed on the dendritic cell, B cells, and other cell population. ICOS play an essential role in T cell dependent antibody response, particularly in the germinal-center reactions. It is required for a development and activation of T follicular helper cells, which are essential for formation of germinal center and for the generation of B cells that produce high affinity antibodies. The outcome of T cell activation is influenced by balance between engagement of activating and inhibitor receptors of the CD28 family. The inhibitor receptor of the CD28 families are CTLA4, cytotoxic T lymphocytes antigen 4, CD152, and PD1, programmed cell death. This is CD279. Their names do not accurately reflect their distribution and function. The main function of this receptor is often described as co-inhibition. to contrast with CD28 and ICOS, whose function is co-stimulation. Note that sometimes CD28 and activation receptor is called co-stimulators. Both inhibitory receptors express the following T cell activation and function limit the immune response. The concept that a balance between activating an inhibitory receptor can control the magnitude of response in the immune system was discussed in chapter 4 in the context of natural killer cells, and figure 4.10. A similar idea is applicable to response of T and the B lymphocyte, although the receptor involved are quite different. Because the physiological function of inhibitory receptors CTLA4 and PD1 is to prevent and control response to self and microbial antigen, and deficiency in their function due to gene mutation or blockade with the antibody causes autoimmune disease. While we will discuss them in more detail in chapter 15 when we consider immunological tolerance and autoimmune. Suffice is to say here that CTLA4 function as a competitive inhibitor of CD28 by binding more strongly to B7 molecules, and PD1 activates a tyrosine phosphate that blocks tyrosine kinase dependent signaling by TCR and CD28. It is likely that various costimulators and inhibitory receptor of the B7 CD28 were family serves distinct role in different immune receptors or at a different stage of response. It is thought that the CD28 ratio B7 interaction is most important for initiating T cells response by activating naive T cells. ICOS and ICOS ligands interaction are critical for helper T cell dependent antibody response. The CTLA4 B7 interaction inhibits the initial activation of the T lymphocyte in secondary lymphoid organ, and PD1 ligands interaction mainly inhibit the activation of effector T cells response to antigens such as CD8 T cells response, especially non-lymphoid tissue. Other costimulatory pathway, many other T cell surface molecules have been shown to deliver costimulatory signals in vitro, but their physiological role is promoting T cell activation is less clear than that of CD28 family. Several putative costimulatory receptors belong to the large tumor necrosis factor receptor superfamily, and their ligands are members of tumor necrosis factor family. Many of these receptors are expressed in activated T cells and regulatory T cells and have been shown to stimulate or inhibit immune response under various experimental conditions. OX40 or CD134 is a tumor family member expressed on the activated CD4 and the CD8 T cell. They function to maintain cell survival and sustain the response. Its ligand, XO40 ligand, is expressed on activated antigen-presenting cells. CD137 and CD27 are two other tumor necrosis factor superfamily molecules that are expressed on activated and memory cell as well as regulatory T cells. Their roles in regulating immune response are not well defined. T cell also expresses numerous inhibitory receptors in addition to CTLA4 and PD1, but their physiological function are also not well established. Two that have received a considerable interest are TIM3 and LAG3 inherited mutation in time associated with systemic inflammation disease. The interaction of CD40 ligands and T cells with CD40 and on antigen enhances the T's response by activating the antigen-presenting cell. CD40 ligands is a tumor necrosis factor superfamily membrane protein that is expressed primarily on activated T cell. And CD40 is a member of tumor necrosis factor superfamily expressed on B cell, macrophage, and dendritic cell. The function of CD40 in activating macrophage is cell-mediated immunity, and activated B cells in humoral immune response are described in Chapter 10 and Chapter 11, respectively. Activated D helper T cell expresses CD40 ligand, which engage CD40 on antigen-presenting cell and activate antigen-presenting cell, making them more potent for enhancing the expression of B7 molecule on the cytokines such as Interleukin-2 that produce a T cell differentiation. This phenomenon is sometimes called licensing, meaning that activated T cell license antigen-presenting cell to become more powerful stimulators of immune response. Thus, the CD40 pathway indirectly amplifies the T cell response by inducing costimulator on antigen-presenting cells, but CD40 ligands does not by itself function as costimulatory T cell. Therapeutic targeting costimulators. Based on the understanding of costimulatory pathway, therapeutic agents have been developed for controlling injuries immune response by inhibiting costimulation called costimulatory blocking. CTLA4 a fusion protein consisting of extracellular domain of CTLA4 and Fc portion of human IgG, bind to B7-1 and B7-2 and block the B7 CD28 interaction. The reason for the use of extracellular domain of CTL4 rather than CD28 to bind to and block this the B7 molecule is that CTLA has higher affinity for BL than does CD28. Attachment of Fc portion of IgG increases the in vivo half-life of proteins. CTL4 Ig is an approved therapy for rheumatoid arthritis and transplant rejection. Inhibitors of CD40 ligands pathway are in clinical trial for transplant rejection and autoimmune disease. Antibodies that block the CTLA4 and PD1 inhibitor receptor are approved for immunotherapy of tumor. They work by preventing CTLA4 and PD1 from binding their ligands, thereby reducing inhibition and thus enhancing T cell activation and enabling the cancer-bearing individual to mount more effective anti-tumor immune response. See Chapter 18. Because cell inhibitor receptor impose checkpoint on immune response, blocking them therapeutically to enhance immune response is called checkpoint blockade. As one might predict from the role of inhibitor receptor in maintaining self-tolerance, blocking them for cancer immunotherapy induces autoimmune reaction in many patients. Functional response of T lymphocyte. Sorry. Functional response of T lymphocyte. The earliest response of antigen-stimulated T cell consists of changes in expression of various surface molecules, including cytokine receptor as well as the secretion of cytokine. These are followed by proliferation of antigen-specific cell driven in part by secreted cytokine, and then differentiation of activated cells into effector and memory cell. In the remainder of this chapter, we will describe these steps, the underlying mechanism, and functional consequences. So, like in this description, very nice diagram, you see there's the dendritic cell, which is antigen presenting cell. We have B7, CD28, then we have T cell. When these ones work together, there is activation. But when we see in costimulatory blockade, we see there is a CTL4IG, and when they bind to T cells, then they can block the costimulatory effects. So let us read a little bit about the instruction there. Figure 9.7 says the mechanism of therapeutic costimulatory blockade A, the normal T cell response induced by antigen recognition and co-stimulation mediated by B7CD28. Then we see in B a fusion protein consisting of extracellular portion of cytotoxic T lymphocyte antigen 4 CTLA and the Fc tail of immunoglobulin G molecule is used to bind and block the it can go and block the B7 molecule, thus preventing the interaction with the activating receptor CD28 and inhibiting T cell activation. So you see here the B7 is already occupied, and also this one is already occupied. So if these are occupied with CTLA, cytotoxic T lymphocyte antigen 4 IG immunoglobulin, then this one gets nothing to bind within. Costimulatory cannot take place, therefore there is blockage, so immune action cannot take place. Very nice diagrams. Changes in surface molecule during T cells activation. After activation by antigen recognition and co-stimulation, there are characteristic changes in expression of various surface molecule in T cells. Many of the molecules that are expressed in activated T cells are also involved in functional response of the cell. CD69. Within a few hours, T cell increases the expression of CD69. This protein binds to and reduce surface expression of sphingosine 1 phosphate receptor, CYP1, which are described in chapter 3 as receptor that mediate egress of T cell from secondary lymphoid organs. The consequences of decreased CYP expression is that activated T cells are retained in lymphoid organs long enough to receive the signal that initiate their proliferation differentiation into effector and memory cell. After that occurs, CD69 expression decreases. The activated T cells re-expresses high level of CYP, and therefore the effector and memory cell can exit to lymphoid organs. Then there is expression of CD27, Interleukin-2 Receptor alpha. The expression of this component of a receptor for growth factor Interleukin-2 enables activated T cells to respond to this cytokine. This process is described later. Then there is CD40 ligands, and which is CD154. Within 24 hours to 48 after antigen recognition, CD4 T cell expresses high level of ligand CD40. The expression of CD40 ligand enables these activated T cells to mediate their cell effector function, which are to help macrophage and B cell. In addition, as discussed earlier, CD40 ligands and T cell activates the dendritic cell to become better antigen presenting cell, thus providing positive feedback mechanism for amplifying the T cell response. Then we have CTL4 and PD1. CTLA4 is expressed on T cell within 24 hours and 48 hours after antigen recognition, and PD1 may be induced even more rapidly. The role of these inhibitors in limiting immune response is described later in the chapter, in self-tolerance in chapter 15, and in tumor immunity chapter 18. So, in reminder, these are proteins that are produced. These are CD4 that are produced, CD69, CD25, which we'll discuss later, CD40 ligand, which are produced within 48 hours from 24, then which is after antigen recognition. CD4 cells expresses high level of ligand 40. This enables the activated T cell to mediate their effector function, but again, we are told the CD40 ligands activate the dendritic cell to become even better presenting cell. But there will be production of inhibitor factors, the CTLA4 and PD1 this time. So if we have to read anything, here we have changes in surface molecule after T cell activation. The approximate kinetic and overexpression of selected molecule during activation of T cells by antigen and costimulators are shown. The illustrative example include a transcription factor, F-phos, cytokine, interleukin, and surface protein. These proteins are typically expressed at low level in naive T cell and are induced by activating signal CTLA4, cytotoxicity, lymphocyte, antigen, cytotoxicity, lymphocyte, antigen 4, and there is PD1, programmed cell death protein 1, induced the hour or 2 days after initial activation. The kinetic estimates and will vary with the nature of antigen in dose and persistence and type of adjuvant. B, the major function selected surface molecule shown and described in the text, CD40 ligands, Interleukin-2 receptor. So let us continue. The other molecules that are produced, they are still discussing the many molecules that are expressed in activated cells. The other one is adhesion molecule and chemokine receptor. During activation, T cell reduces expression of molecules that bring them to secondary lymphoid organs, such as L-lectin, that is CD62L, and the chemokine receptor, CCR7, and increases the expression of molecules that are involved in their migration to peripheral sites of infection or tissue injury, such as integrin, LFA1 and VL4, and ligand for E and P selectin, and various chemokine receptor. These molecules and their roles in T cell migration were described in chapter 3. Activation also increases the expression of CD44, a receptor for extracellular matrix molecule hyaluron, binding for CD44 to its ligand to help retain effector T cells in tissue as site of infection and tissue damage. Cytokines in T cell activation. Numerous cytokines plays critical roles in adaptive immune response. CD4 help a T cell make the largest amount of variety of these cytokines, but some are also produced by CD8 T cells and antigen presenting cell. Cytokines secreted by dendritic cell and other antigen-presenting cell are especially important for differentiation of naive T cell into different types of effector cells. Various cytokines are involved in the proliferation and differentiation of antigen-stimulated T cells and in effector function of these cells. Most of these cytokines act on the cell that produce them, autocrine action, or nearby cell, paracrine action. The role of cytokine in effector function of T cells are described in Chapter 10 and Chapter 11. Here we discuss Interleukin-2, the prototype of T cell-derived cytokine that stimulates T cells' response. So let us discuss Interleukin-2 secretion and Interleukin-2 receptor expressions. Interleukin-2 is a growth, survival, and differentiation factor for T lymphocytes that play a major role in proliferation of antigen-stimulated T cell and in maintenance of functional regulator T cell. So, I repeat again, Interleukin-2 is a growth, survival, and proliferation factor. A growth, survival, and proliferation factor for T lymphocytes that play a major roles in the proliferation of antigen-stimulated T cells and in the maintenance of functional regulatory T cell. Interleukin-2 acts on the same cells that produce it and on the adjacent cell. This is function as autocrine or paracrine cytokines. Interleukin-2 is produced mainly by CD4 T lymphocytes rapidly after recognition of antigen and costimulators. Activation of T cells stimulate transcription of interleukin-2 gene and the synthesis and secretion of protein. Interleukin-2 production is rapid and transient, starting within 1 to 2 hours after antigen recognition, picking up to 8 to 12 hours and declining by 24 hours. Secreted interleukin-2 is the 14 to 17 kilodalton globular proteins containing 4 alpha helices. It is the prototype of the 4 alpha helical cytokines family that interact with the type 1 cytokines receptor, seen in Chapter 7. The high affinity of interleukin-2 receptor is transiently expressed on the activation of naive and effector T cells. Regulatory T cells also express this receptor. The interleukin-2 receptor consists of 3 non-covalent associated proteins, the interleukin-2 Ra-alpha, CD25, interleukin-2 15-RB, CD122, and gamma C, CD132. Of the 3 chains, only interleukin receptor alpha is unique to interleukin receptor. So the beta chain is also part of interleukin-15 receptor. The gamma chain is shared with number of cytokine receptor, including those for interleukin-4, 7, 9, 15, and 21. And it is therefore called the common gamma chain, YC. Both the B and gamma C chain engage in Janus kinase, JAK and STAT signaling, transducers, and activation of transcription signaling pathway. Interleukin-2 receptor beta gamma C complexes are expressed at low level on the resting T cells and on natural killer cell, and bind to interleukin-2 with equilibrium dissociation constant approximately 10 power negative 9. On activation of naive CD4, CD8 T cell, the alpha chains associate with the B gamma C complex to form the completely Interleukin-2 receptor and Interleukin-2 RB complex, which can bind to Interleukin-2 more tightly with a dissociation constant of approximately 10 power 11 Growth stimulation of activated T cell occur at a similarly low interleukin-2 concentration. Because both interleukin-2 secretion and interleukin-2 receptor production occur in response to antigen stimulation, the antigen-activated T cell are the ones that proliferate preferentially in the response to the cytokines, compared with the bystander cell that have not recognized the antigen. Interleukin-2 produced in response to antigen stimulation. It is itself stimulant of induction of interleukin-2 receptor, providing a feedback mechanism by which T cell response amplify themselves. Activated T cell expresses interleukin-2 receptors transiently long enough to respond to the growth factor and proliferate. CD4 irregulatory T cells expresses a completely interleukin-2 receptor complex and require 2 interleukin for the maintenance. As discussed in the following section and in the chapter 15, chronic T cell stimulation leads to the shedding of interleukin-2 receptor, and an increased level of shed of interleukin-2 receptor in the serum is the mark of strong antigenic stimulation. This is acute rejection transplanted organs. Function of interleukin-2. The biology of interleukin-2 is fascinating because it plays critical role in both promoting and controlling T cell response and function. Interleukin-2 stimulates the survival, proliferation, and differentiation of antigen-activated T cells. Interleukin-2 promotes survival of the cell by inducing the anti-apoptotic protein BCL2. It stimulated cell cycle progression through activation of mTOR, mechanical surrogate of rapamycin, which induce the synthesis of cyclin and relieve block in the cell cycle progression through degradation of the cell cycle inhibition. In addition, interleukin-2 increases the production of effector cytokine. This is interferon gamma, interleukin-4 by T cell. Interleukin-2 is required for the survival and function of regulatory T cell, which suppress the immune response against self and other antigen. These cells constitutively express complete interleukin-2 receptor, including the chain of CD25, are much more sensitive for interleukin-2 than activated effector T cell. We'll discuss this role of interleukin-2 in more detail in chapter 15 when we describe the properties and function of regulatory T cells. An interesting feature of this function of interleukin-2 is that regulatory T cells do not produce cytokines, so they depend on interleukin-2 made by other T cells responding to antigen. Clonal expansion of T cells. T cell proliferation in response to antigen recognition is mediated by a combination of signals from antigen receptor, costimulators, and the autocrine growth factor, primarily interleukin-2. The expansion of antigen-specific clone that results from this proliferation converts a small pool of naive antigen-specific lymphocyte into large number of cells required to eliminate antigen. Before antigen exposure to frequency of naive T cells specific for any antigen is 1 in 10 power 5 to 10 power 6 lymphocyte or fewer. After microbial antigen exposure, the frequency of CD8 T cells specific for that microbes may increase to as many as 1 in 3 CD8 T cell lymphocyte, representing a great number of 50,000-fold expansion of antigen-specific CD8 T cell and the number of specific CD4 cell increases up to 1 in 100 CD4 lymphocytes and 1,000-fold expansion. Studies in mice first showed this tremendous expansion of antigen-specific population in some acute viral infection, and remarkably, it occurred within a little as 1 week after infection. Equally remarkable was finding that during this massive expansion of antigen-specific clones, bystander T cells that not specific for virus disease did not proliferate. The expansion of the T cell specific for Epstein-Barr virus and human immunodeficiency virus in acutely infected human is also on this is also on this is also on this order of magnitude, is also on this order of magnitude. So you can go through the diagrams, they are very nice ones. So T cell activation by antigen and costimulators happen there. Then secretion of interleukin-2 when T cell, the main, especially T helper, main producer of interleukin-2. Then interleukin-2, if it is produced, it can auto-react, it can auto-signal. So expression of interleukin-2 receptor chain formation, high affinity, complex, and the interleukin-2 induced T cell proliferation, to proliferate, increase in number. So regulation of interleukin-2 receptor expression of resting naive T cell lymphocytes expresses the interleukin-2 gamma YC complex, which has a moderate affinity for interleukin-2. Activation of T cell by antigen costimulators and interleukin-2 itself leads the expression of interleukin-2 receptor chains, alpha chain, called the CD25, and increase the level of high affinity interleukin-2 complex, interleukin-2. So here we see the dendritic cell when it goes, that's active as antigen presenting cells. Then with costimulator, what happens? Naive T cells is activated. After activated, you get effector cells. Proliferation and differentiation of effector and memory T cell happen. Then it be the dendritic cell there. Then activation happen. Interleukin-2 produced. Then they act on regulatory T cell. Regulatory T cell is proliferating. So a biological action of interleukin-2A, interleukin-2 stimulates the survival, proliferation, and differentiation of T lymphocyte, acting as autocrine growth factor, leading to the generation of effector and memory cell. B, interleukin-2 also promote the survival of regulatory T cell and maintain their function capability, and thus control immune response, this against self-antigen. So we saw the diagram already. Then you see this diagram here. This is clonal expansion of T cell. The number of CD4 and CD8 T cell, number of CD4 and CD8 T cell. So the number of repeat here. The number of CD4 and CD8 T cells specific for microbial antigen and expansion and decline of cell during immune response are illustrated. The number approximation based on the studies of model microbial and other antigens in the inbred mice. The relative number of viable infectious organisms in individual over time is indicated by tangent shared curves. Okay, so contraction of homeostasis, clonal expansion. You have infection goes, they expand. Differentiation of activated T cell into effector cells. Many of the progens of antigen-stimulated T cells differentiate into effector cells. As summarized in the overview of chapter, effector cell of CD4 lineage expresses surface molecule and secretes cytokines that activate the other cells, the B lymphocyte, macrophage, and dendritic cell. Whereas naive CD4 T cells produces mostly interleukin-2 activation, effector CD4 T cells are capable of producing a large number of variety of cytokines that have diverse biological activities. Effector CD8 T cells are cytotoxic and kill infected cells. Because there are important difference in effector cells of CD4 and CD8 lineage, we will describe their development and function separately in chapter 10 and chapter 11. Development and properties of memory T cells. T cell mediated immune responses to an antigen usually result in generation of memory T cells specific for antigen, which may persist for years and even lifetime. Memory cells provide effective defense against pathogens that are prevalent in the environment and may be repeatedly encountered. Despite the importance of immunological memory, many fundamental questions about generation and maintenance of memory cell have still not been answered. Memory cell may develop from effector cell along a linear pathway, or effector and memory cell population follow divergent differentiation and are two alternative fate of lymphocyte activated by antigen and other stimulus. The mechanism that determine whether an individual antigen stimulated T cell will become short-lived effector cells or enter the long-lived memory cell pools are not fully established. However, as effector T cells contract, a small pool of memory precursor effector cell, often called MPEC, develop from which memory population are mainly generated. The signal that drive the development of memory cell are also not fully understood. These signals may include a strength of T cell receptor stimulation, the level of costimulation, the cytokine environment, and others. No single transcription factor determines whether an antigen-stimulated T cells will become a terminal effector cells or a memory cells. Rather, this choice may be controlled by quantitative difference in numerous transcriptional factors and epigenetic reprogramming. Properties of memory T cells. The defined properties of memory cells are their ability to survive for prolonged period after antigen is eliminated and to mount larger and more rapid response to antigen than do naive T cells. Several features of memory cells account for these properties. Memory cells expresses increased level of anti-apoptotic proteins, which may be responsible for their prolonged survival, whereas naive T cell lives for weeks or months and are replaced by mature cells that develop in late thermos. Memory T cells may survive for years. Thus, a human age in an environment in which they are constantly exposed and responding to infectious agents, the proportion of memory cell induced by these microbes compared with naive cell progressively increases. In individuals older than 50 years, half or more of circulating T cell may be memory cells. The anti-apoptotic protein that promote memory cell survival include BCL2 and BCLXL, which block apoptosis induced by deficiency of survival signals. The presence of these proteins allow memory cell to survive even after antigen is eliminated and innate immune response have subsided when the stimulus for effector T cells survival and proliferation are no longer present. So we are looking here. The third characteristic is memory cells respond more rapidly to antigen stimulation than do naive cells specific for same antigen. For example, studies in mice have shown that naive T cells differentiate into effector cells in response to antigen in 5 to 7 days, but memory cells acquire effector function within 1 to 3 days. This is one reason why secondary response to antigen exposure are more rapid than primary response. A possible explanation for this accelerated response is that the gene lock for cytokines and other effector molecules are fixed in an accessible chromatin state in the memory cells, in part because of the changes in methylation and acetylation of histone. These epigenetically modified genes are poisoned to respond rapidly to antigen challenge. Also, the number of memory T cells specific for any antigen is greater than the number of naive cells specific for the same antigen. As discussed earlier, proliferation leads to large clonal expansion in all adaptive immune responses, and the memory cells that remain from an expanded clone are typically 10 to 100-fold more numerous than pool of naive cells before antigen encounter. The increased clonal size is one reason that antigen challenge in the previously immunized individual induces large response than first immunization in a naive individual. Memory cells are able to migrate to peripheral tissues and respond to antigen at these sites. As discussed in Chapter 3, naive T cells migrate preferentially to secondary lymphoid organ where they respond to antigen for the first time, but memory cells can migrate to virtually any tissue. These differences are called to virtually any tissue. These differences are related to difference in the expression, realization molecule, and chemokine receptor. In addition, memory T cells are less dependent on costimulation than naive T cells, allowing memory cells to respond to antigen presented by a wide range of antigen-presenting cells in peripheral tissue. In contrast, as we have discussed earlier, naive T cells are dependent on antigen presenting by mature dendritic cells in secondary lymphoid organ. Memory cells undergo slow proliferation, and this ability to self-renew may contribute to the long lifespan of memory. Then maintenance of the memory cell, long lifespan of memory. The cycling of these cells may be driven by cytokines because the capacity of self-renewal memory cells have been linked to stem cells. Although they survive for long period, memory cells are functional inactive and to be restimulated by antigen and to become functional effector cells. Then we have maintenance of memory cell is dependent on cytokine but does not require antigen recognition. Most important cytokine for maintenance of memory CD4, CD8 T cells is interleukin-7, which also play a key role in early phenotype development. And in the survival of naive T cells, predictably high expression of the interleukin-7, CD127 is characteristics of memory T cells. Memory CD8 T cells also prepares on the related cytokine interleukin-15 for survival. Interleukin-7 and 15 induces expression of anti-apoptotic protein and stimulated low level proliferation, both of which maintain a population of memory of memory T cells for growth period. The ability of memory cells to survive without antigen recognition has been best described by experiment in mice in which antigen receptor are genetically deleted after mature lymphocytes have developed. In these mice, the number of naive lymphocytes dropped rapidly, but memory cells are maintained. So the most reliable phenotypic markers for memory T cell appear to be on surface expression of interleukin-2 receptor and protein of unknown function called CD27 on absence of marker of naive recently activated T cell. See table 2.5. In human, most naive T cell expresses 200 kappa D isoform on the surface molecule, CD45 or CD45R for restricted A, and most memory T cells expresses 180 kD isoform CD45 called RRO, and both CD4 and the CD8 memory T cell are heterozygous and can be subdivided into subsets based on their homing properties and function. Three main subset of memory T cell are known. There is central memory T cell expresses chemokine receptor CCR7 and adhesion molecule L-lectin and home mainly to lymph node. They have a limited capacity to perform effector function when they encounter antigen, but they undergo brisk proliferation response and generate mirror effector cells to antigen challenge. They provide a pool of memory cell that can respond to antigen challenge. And develop in effector cell. Effector memory T cells, on the other hand, do not express CCR7 or L-lectin, and they home peripheral site, especially mucosal tissue. On stimulation by antigen, the T effector memory cell rapidly produce effector cytokines such as Interferon Gamma and become cytotoxic, but they do not proliferate much. This effector subset, therefore, is poised for rapid response to expose to microbes, but complete eradication of its infection may also require a large number of effector generated from the pool of central memory T cell, a subset of T effector cells in human expression CD45 isoform, which is characteristic of naive T cell. This population is called the TAMRA cell, the T effector memory T cell, and they can also unique function or properties not known. We have tissue resident memory T cells. These are present in various non-lymphoid tissue, do not circulate in the blood, and may provide a rapid defense against the microbes in the tissue. So most of these expresses high level of CD69, a molecule that reduces expression of Syllable. As a result, these cells do not respond to high concentration of Syllable in lymph and blood, facilitating their retention in tissue. A fourth population of memory T cells called peripheral memory T cell has been described. These cells make important contribution to the secondary response in tissue, and unlike T cells are capable of moving between circulation tissue. So in summary, what we did here is we have three major subset of memory T cells. Central memory T cell, this is expressed as chemokine receptor CCR7 and adhesion molecule L-lectins. They have limited capacity to perform effector function. When they encounter antigen, but they undergo risky proliferative response to generate many effector cells as antigen challenge. This is central memory cell. Then we have effector memory cell, on the other hand, that doesn't express the CCR7 and lectin. We have tissue resident memory T cells, but currently we have also another type of cells, fourth population memory T cells called the peripheral memory T cell. Memory T cells are also, I'm now there, memory T cells are also here. Memory T cells are also heterozygous in terms of cytokine profile. For example, some CD4 memory T cell may be derived from activated T cells that are not committed to T helper 1, T helper 2, T helper 17 phenotype described, and when reactivated by exposure to antigen and cytokine, they differentiate into any of these subsets. Other memory T cells may be derived from differentiating T helper 1, T helper 2, and T helper 17 effector and retain their respective cytokine profiling on reactivation. Now, decline of T cell response. Elimination of antigen leads to the contraction of T cell response, and this decline is responsible for maintaining homeostasis in the immune system. Several mechanisms may account for the decline. First, cell death. As antigen is eliminated, an innate immune response associated with antigen exposure abates. The signal that is normal keep the activated lymphocyte alive when proliferate and are no longer present. As mentioned earlier, co-stimulation and growth factor such as interleukin-2 stimulate expression of anti-apoptotic proteins BCL2 and BCLXL in the activated lymphocyte, and these proteins keep cell viable. As the level of costimulatory and amount available of interleukin-2 decreases, the level of anti-apoptotic protein in the cell drop. At the same time, growth factor deprivation activates sensor of cellular stress such as BH3 only protein beam, which trigger the mitochondrial pathway of apoptotic and are no longer opposed by anti-apoptotic protein. The net result of these changes is that most of the cells that were produced by activation die on the generation of new activated cell decline, so the pool of antigen-activated lymphocytes contract. Secondly, mechanism where cell decline is inhibition of T cell activation. A number of mechanisms inhibit T cell response after they have been initiated. The mechanisms likely contribute to the physiological downregulation of immune response to pathogen, and they also are reportedly regulators of self-tolerance. So there is a reduced activating signal. Several intracellular enzymes function to limit the duration or magnitude of T cell activation response. These include phosphatases such as SHP1, SHP2, SHIP, which dysphosphorate signaling substrate, and E3 ubiquitin ligase such as CBLB, which degrade activated signal intermediates. These enzymes may be activated after T cell receptor and costimulatory pathway are engaged. Other one is engagement of cell surface inhibitory molecules as PD1 and CTLA4 are expressed in activated lymphocyte, and both may function to limit the continued activation. CTLA upregulation on recently activated T cell may primarily inhibit T cell activation in lymph nodes by competing with the CD28 B7 protein and thus prevent the generation of effector memory cells. PD-1 is induced in both activated CD8 and CD4. Its inhibitory function is best understood in the context of restricting previously activated CD4, CD8 T cells in the peripheral tissue. It is not clear if these co-inhibitor regulators regulate all the T cell response and or response mainly selected antigen, some virus, tumor, and some antigen. Regulatory T cells. Most immune response in which effector T cells are activated are accompanied by the development and activation of regulatory T cells. Regulatory T cells function in several ways to inhibit T cell response, including the engagement of CTLA4 on their surface with B7 and on antigen presenting cell, as discussed further in chapter 15. Regulatory T cell limit immunopathology during infection and also suppresses immune and allergies. Summary. So, T cell response are initiated by signal that generated by T cell receptor. Recognition of peptide major histocompatibility complex on the surface of antigen presenting cells and through signal provided at the same time by costimulator expressed on the antigen presenting cell. The best defined costimulator are members of the B7 family, which are recognized by receptors of the CD28 family expressed on CD or on T cells. Expression of B7 costimulator on the antigen presenting is increased by encounter with microbes, providing a mechanism for generating optimal response against infectious pathogen. Some members of the CD28 family inhibit T cell response, and the outcome of T cell antigen recognition is determined by the balance between engagement of activating and inhibitory receptor of this family. T cell response to antigen and costimulator include changes in expression of surface molecule, synthesis of cytokine and cytokine receptor, cellular proliferation and differentiation into effector and memory cells. The surface molecule whose expression is induced by T cell activation include proteins that are involved in retention of T cells in lymphoid organs. Cytokine and cytokine receptor, effector and regulatory molecule, and molecules that influence antigen, that influence migration of the T cell, not antigen. Shortly after activation, T cell produces So the T cell's response to antigen and costimulator include changes in expression of surface molecule, synthesis of cytokine and cytokines receptor, cellular proliferation and differentiation into effector and memory cell. The surface molecule whose expression is induced on the T cell activation include proteins that are involved in retention of T cell in lymphoid organ, cytokines and cytokine receptor, effectors and regulatory molecules, and molecules that influence the migration of T cell. So shortly after activation, T cell produces cytokines, Interleukin-2, and expresses high level of functional Interleukin-2 receptor. Interleukin-2 receptor drive the proliferation of cell, which can result in marked expansion of specific clones. Same activated T cell may differentiate into memory cell, which survive for long period and respond rapidly. The the maintenance of memory cell is dependent on cytokines such as interleukin-7, which may promote the expression of anti-apoptotic proteins and stimulate low-level cycling. Memory T cells are the heterozygous and consist of the population that differ in migration properties and functional response. So T cell responses decline after elimination of antigen, thus returning the system to rest. The decline is because signal for continued lymphocyte activation and eliminated and become virus historical mechanism. And because virus inhibitory mechanism. So these are a number of selected reading, T cell activation, and many other things. And this marks the end of the chapter. Get prepared for next chapter, which is differentiation and function of CD4 effector T cell. Very nice topic. So get prepared. End up here today.